My name is Krisha Anand, and in this video series today, we'll be talking about how to break down research papers. It can often be complicated to get through one of these just because they're really long or because they have complicated jargon, but together we will go through every single word in this paper and analyze what it means and how to distill it down to the main points. If you like this video, give it a thumbs up below and subscribe to my channel, and let's get started. Okay, so this is our first article. I got this from NCBI, which is a free database online, so you can get a lot of free research articles on that. And I can post the link to this one in the description, so you can check it out there as well. So when I was looking for this article, actually, I was trying to find something that might be easy enough to, share with everyone, even people who may not have a background in biology, but also something that's difficult enough that everybody gets to learn something new. And I came across this particular article. I was kind of reading through the article and just kind of skimming through it, looking at the images, all that. And it seemed, pretty doable, like not overly complicated. And then I just went to check out the length. So I copy-pasted the article into a Google Doc, and it was 23 pages long. So yeah, this one is going to be a long one. I might split this up into 2 videos, so we'll see how that happens. Yeah, so let's get started then. So the title is Cell Extrusion, a stress-responsive force for good or evil in epithelial homeostasis. So I think the first question in everybody's minds is, what is cell extrusion? And I'll admit, when I started reading this, I had no idea either. But if you look at the title itself, they do kind of describe what it could be. It is a stress-responsive force for good or evil in epithelial homeostasis. So the epithelium is basically a tissue lining of any hollow organs or body surfaces. And homeostasis refers to a condition being maintained, essentially. So some condition in the epithelium is being maintained through cell extrusion, and cell extrusion is caused by a stress-responsive force. Okay, so far that makes sense. But again, not too detailed, and we still don't entirely know what cell extrusion is. So let's go to the abstract for that. In the abstract, it goes right into saying epithelial tissues robustly respond to internal and external stressors via dynamic cellular rearrangements. So what do we get from this? This tells us much the same thing that we found in the article title, that there are internal and external stressors that affect epithelial homeostasis, presumably through cell extrusion. And indeed, like the next sentence does confirm that cell extrusion acts as a key regulator of epithelial homeostasis. And then it gives us 3 mechanisms for how this happens: by removing apoptotic cells, orchestrating morphogenesis, and mediating competitive cellular battles during tumorigenesis. So what does that mean? Well, apoptosis is essentially a form of cell death, so they just combust. And cell extrusion helps remove those cells. It also orchestrates morphogenesis. This is actually a pretty easy word to understand if you just split it up into 2 parts. Morpho is shape, and genesis is to create. So essentially, cell extrusion helps to create the cell shape. And it also mediates competitive cellular battles during tumorigenesis. And as we just said, genesis is to create. So it prevents the formation of tumors. Great. So stops cancer, basically, or at least helps to prevent it. And then They go on to say that cell extrusion plays critical roles during development and disease, or has diverse functions in development and disease. And another important point that they make in the abstract here that we should pay attention to is that there's a strong intersection between cell extrusion and cell competition, and that these two forces, essentially, mechanisms, work together to suppress tumors. And if either one is not regulated properly, then we end up with cancer. Okay, so just by reading the abstract, we got a lot there. We got some of the functions of cell extrusion, and we kind of understood why this is so important in the body. And more than that, we finally got our definition of what cell extrusion is. So cell extrusion, we now know, helps to maintain homeostasis in epithelial tissue by acting as a form of regulation and essentially removing unnecessary cells when there is overcrowding, which is often like one of the signs of like tumors building or one of the reasons that apoptosis might occur. And it responds to essentially stressors like crowding and other internal and external stressors. Okay, so now we can go on to the first section and really get into the meat of this article. So it says here that cell extrusion is a force for good, and that is like the first half of this article. It talks about the role of cell extrusion in development and tissue homeostasis. So what's going on here? Complex cell rearrangements are required for shaping tissue and organ structures in normal development. During such processes, cell extrusion actively regulates development and enforces homeostasis by expelling cells from crowded regions, initiating cell differentiation and epithelial mesenchymal transitions, and exerting physical morphogenetic forces. Okay, again, the first sentence there is just saying that different cell rearrangements are needed for normal tissue and organ development, and that cell extrusion plays certain roles in this. It enforces homeostasis by expelling cells from crowded regions, which we kind of established in the abstract itself since it helps in apoptosis and in tumorigenesis, and plays a role in tumorigenesis anyway. And we also get here that it initiates cell differentiation. So in cell differentiation, you essentially go from a stem cell that has no specific function and can kind of turn into many different types of cells. And you go from that, like a baby cell basically, to a fully matured cell that has a more specific function. And another function is that it initiates epithelial mesenchymal transitions, EMTs. So when I picked up this article, I'd never heard this term before, so that's actually one of my favorite part about reading research papers. You get to hear all these new terms and learn all these new concepts, and it's super exciting. So yeah, I looked this up immediately, and again, if you break it down into its different parts, it's pretty easy to understand. We already understood what the epithelium was before, which we defined in the article title itself, actually. And then now we need to understand what are mesenchymal. What does mesenchymal mean? So mesenchymal cells are cells that can differentiate into connective tissue, lymphatic tissue, or blood vessels. So the epithelial mesenchymal transition is basically a process where an epithelial cell turns into a mesenchymal cell. That's a more simplistic way to put it. And we also know from this little paragraph that cell extrusion exerts morphogenetic forces. Again, we looked at what morphogenesis was in the abstract. So yeah, that's 2 paragraphs in and we're doing pretty good. Okay, so now our next section is our longest one yet. We have the topic cell extrusion relieves overcrowded tissues. So it basically talks about one of the functions of cell extrusion, goes more into depth about that. And if we take a look at this, it has 1, 2, 3 paragraphs. The thing is, there is also a picture here. You have 2 diagrams. And when you get diagrams in a section, you definitely want to go over the diagrams first, because they'll cover a lot of info that's also in the text. But it'll be a lot easier to understand just because it's in the format of a diagram. So let's go ahead and read this caption first. In response to apoptotic stress, cells undergoing apoptosis produce S1P via SPHK. Both of these are basically proteins, which bind to S1P receptor, S1P2, in neighboring cells. S1P2 activates Rho signaling through this specific protein, essentially activating the Rho pathway, recruited basally by microtubules, triggering basal actomycin contraction and subsequent apical extrusion of the dying cell. So taking a look at this diagram, let's dissect what that really means. So you start out with the SPHK protein, which then activates the S1P protein. S1P then goes ahead and binds to S1P2 receptor, and this kicks off the Rho pathway, as you see here. And the Rho pathway then leads to actin and myosin contraction. And this produces this apical extrusion by creating this force, as we saw here in the title, this stress responsive force that they were talking about. And okay, that makes sense. But that is for apoptotic cell extrusion. The thing is, there's also live cell extrusion, which is caused by crowding stress instead of apoptotic stress. So how is that different? Well, as we can get from this second caption, part B, over here, it's not actually that different. If you look at the diagram, you still have SPHK, you still have S1P and S1P2, you still have the Rho pathway and actin myosin. Really the only thing that's different as a result of crowding stress is this protein right here, piezo1. What is the function of piezo1? So, shown by the caption, we do know that piezo1 triggers live cell extrusion and basically triggers SPHK and the S1P Rho pathway. But the exact mechanism through which this occurs is not yet known. So yeah, unfortunately that's still a mystery, but we do know this pathway happens in this form. Okay, so we can now go to the actual text itself, and it's going to be a lot easier to understand now since we've got the gist through the diagrams. So it starts off by saying that epithelia can remove surplus cells from overcrowded regions by extruding live or dying cells. So essentially, if it's extruding live cells, then you have live cell extrusion, which was shown in diagram B here, due to crowding stress. Whereas if you have dying cells, we can refer to diagram A, and you have apoptotic cell extrusion. So they're basically 2 different mechanisms by which this happens. Furthermore, we know that crowding-induced cell extrusion occurs in diverse tissues and cell cultures, including human colon epithelia, zebrafish epidermis, the Drosophila pupilnatum, and Madden-Darby canine kidney monolayers, or MDCK cells. This sentence basically just gives us a brief overview of all the different types of cells that cell extrusion can happen in. But more importantly, modulating cell growth and density is sufficient to alter extrusion rates, suggesting that overcrowding-induced mechanical forces trigger extrusion. So this actually makes a lot of sense because, as we were saying before, one of the purposes of cell extrusion as a mechanism for maintaining epithelial homeostasis is preventing overcrowding and also playing a role in tumorigenesis. So if you're trying to avoid the growth of tumors, for instance, it makes sense that you would want a mechanism that responds to overcrowding and excessive growth. For example, if you had really dense crowding of cells or like really rapid cell growth, then you would want more cell extrusion because you want to get rid of those cells, get rid of like the excess cells. You also get from this paragraph that in crowded regions, stochastic cell anisotropy may promote topological rearrangements of cell-cell boundaries to promote extrusion, as geometrically induced topological defects are sufficient to extrude MDCK cells. Okay, so we need to define a couple of words here. First of all, what are MDCK cells? As we saw before, these are the canine kidney cells. So again, they're just giving an example of a type of cell in which this overcrowding stress happened. And the other word or phrase we need to really describe here is stochastic cell anisotropy. So what is that? I looked this up because I didn't know either, and I couldn't actually find a good definition of this phrase. But if you break this down into its parts, it's quite easy to understand. Starting with the first word, stochastic means random, and anisotropy means to grow in different directions. So together you have stochastic cell anisotropy is random growth in different directions. Kind of to visualize this, one thing that I found that was useful was imagining a plant. When you have a plant, it's not like all the branches grow exactly the same length or the same direction. You have a lot of different branches that are different lengths, and some might grow upwards, some might like droop down a little bit maybe, and it's all kind of random. You don't necessarily know where a particular branch is going to be. So it's much the same with cells. So in crowded regions, when you have like stochastic cell anisotropy, so random cellular growth in different directions, you want cellular extrusion because this kind of cell growth may not be regulated. So you need to get rid of that. Okay, so we got through this first paragraph, and that makes sense now. Essentially, in this paragraph, they were just talking about the fact that cell extrusion plays a role in modulating cell growth and density. And they were kind of giving the reasons for why and how this happens. The second paragraph here, we actually don't need to go over because this is what we just looked at in these two diagrams. It actually covers the exact same information. So, as I said, looking at the diagrams first definitely makes the article a lot easier to get through because going through all this text does not sound really fun to me. And yeah, going through the diagrams first can make it a lot easier so you can get to the interesting points really quickly. So yeah, let's move on to the third paragraph. Notably, although blocking crowding-induced extrusion can cause atypical cellular accumulations, such as in S1P2 deficient zebrafish, organismal-wide consequences of preventing crowding-induced extrusion reported thus far appear relatively mild. For example, blocking midline notum extrusion merely caused wider adult thorax midlines. The most compelling case for a key role for live cell extrusion in maintaining organismal homeostasis is in mouse secondary palate development and fusion. Okay, let's just look at this part real quick. They say that in certain organisms and in certain experiments, such as with midline notum extrusion, there wasn't really a strong case for why cell extrusion is necessary in the first place, because there weren't really super severe effects of not having it. But then they do say that there was a compelling case for it in mouse secondary palate development. So in the rest of the sentence, they just kind of describe how this experiment was conducted in mice, and in this last sentence of the paragraph, they summarize the results, saying that, nevertheless, the conservation of crowding-induced extrusion between diverse tissues and cell cultures implies that such extrusion is important for stressed epithelia, and thus likely confers organismal benefits. Essentially, as we previously kind of figured out from the diagrams and from the first two paragraphs, the conservation of crowding-induced extrusion essentially helps to ease stressed epithelium, and this helps the entire organism. So there is, in fact, a reason that cell extrusion might be necessary in the body to stop overcrowding and to make sure that the density of cells in any epithelium in the body are, kept at a constant condition, kept at their normal rates, and maintained via homeostasis. And that is what we get from this section here. And as we saw in these diagrams and in paragraph 2, this is modulated by the S1P Rho pathways. And for live cell extrusion, perhaps also through PSO1, though we don't exactly know how that plays a role. Cool, so we have just finished a huge section in this article. So great job for sticking through that, and let's go on to the next one. Now let's move on to yet another function of cell extrusion. Just scrolling through this section again of how cell extrusion couples cell location to cell fate, we see that there is in fact another diagram here. So let's take a look at this. What this diagram essentially shows us in Drosophila neuroblasts is that certain proteins like Baz, actomyosin, and INSE and PINs localize to certain regions of an apoptotic cell. Essentially, what's happening here is that these proteins go to the apex and others like NUM go to the base. And these two regions of the apoptotic cell are determined by the actomyosin, which actually comes from the mother's epithelium, as shown here. And this localization causes delamination of the cells. And as we know, delamination is a process by which the cells form different layers and then differentiate. So in this case, since these are neuroblasts, they would differentiate into either neurons or glial cells. So let's see how that links to cell extrusion. Reading through this first paragraph, we see that it describes much of what we saw in the diagram, but it also adds a couple of other things. It says that neuroblast gene expression is linked to extrusion timing. For example, the key determinant INSC becomes apically localized during the delamination process. Blocking neuroepithelial cell delamination by overexpressing the Notch intracellular domain compromises neurogenesis and is lethal. So, what is the Notch intracellular domain? Essentially, Notch signaling is a way of regulating stem cell maintenance, and the intracellular domain activates the expression of certain target genes. So if you have an overexpression of the intracellular domain, then it's not properly signaling which genes should be activated and which should not. So again, that would block neuroepithelial cell delamination. But the main point of this paragraph is essentially that cell extrusion and cell fate are linked by the expression of actomyosin. As we saw before, actin and myosin help to trigger, as we saw in this diagram, apical extrusion. So they are extremely important in that process and also play a role here in delamination. So thus, cell extrusion and cell location are coupled with cell fate. We also have a few more examples of that in these paragraphs. Looking at the first one here, neuroblast delamination is driven by a pulsatile actomyosin network localized apically in neuroblast precursors, with Rho signaling important for medial myosin accumulation. Again, what we see from this paragraph, and what they go on to say as well, is that Rho signaling, since the Rho signaling pathway is what activates the actomyosin signaling, Rho signaling also plays a role in linking cell fate and cell extrusion. Essentially, they go on to say this is in contrast to removal of apoptotic or live cells by non-autonomous Rho signaling. And they also say that apoptotic cells require autonomous actomyosin force, and the cortical actomyosin ring formed in dying cells is required for neighboring cells to form rosettes around apoptotic cells prior to assembling the multicellular actomyosin ring. Yeah, so basically the main point of that, as we said, was that both actomyosin and Rho signaling play important roles in linking cell fate to cell extrusion. I don't know if it's just me, but I think the concept is really cool because cell extrusion takes away dying cells. It plays a role in apoptosis, and determining cell fate and delamination helps to, kind of set the role of new cells. So it goes both ways. These proteins really do play a role in both the creation and the new purpose of cells, as well as the destruction of cells. The second paragraph here says that conceptually similar to neuroblast delamination, embryonic neural crest cells, morphogenesis in vertebrates, involves NCC delamination from the dorsal neural epithelia, followed by characteristic EMT. Again, we see here that actually we first need to define neural crest cells, and these are basically cells that later differentiate into like peripheral nervous system cells or into craniofacial cartilage or bones, and also various other types of cells, but these are the main ones. What they say here is that these crest cells, their morphogenesis and delamination is followed by a characteristic EMT. EMT, as we saw before, was the epithelial mesenchymal transition. And actomyosin and Rho signaling are again important for orchestrating extrusion, with Rho being required in chick NCC delamination. And Rho-associated protein kinase and myosin required for zebrafish NCC delamination. So basically, again, they play a role in this migration of the NCC cells through EMT, and thus help in delamination. and yet they also play a role in cell extrusion that we previously visited in the previous section. The last example talks about lung development. And here we see that lung neuroendocrine progenitors are selected from bronchial epithelia by an extrusive event, with random cell subpopulations extruding and undertaking an EMT-like migration before settling at lung branch points as committed neuroendocrine progenitors. Cell extrusion thus appears situated as a key mechanism for initiating migration and differentiation during development. Again, primarily through the actomyosin and Rho signaling pathways. This EMT-like migration can occur for these lung neuroendocrine progenitor cells as they go to lung branch points. And it is mediated by many of the same proteins and signaling pathways that are also involved in cell extrusion. So in these ways, these two processes are very linked, and cell extrusion therefore plays a role in determining cell fate as well. And the next section is cell extrusion generates morphogenetic forces. Again, as we talked about before, genesis is to create and morpho is shape. So essentially, cell extrusion helps to mediate how different tissues are sculpted. And that's essentially what the first paragraph here tells us. And yeah, we basically get from this that apoptotic cell extrusion can exert mechanical morphogenetic forces. And now that we know that this occurs, let's go to the diagram, because there is also one included for this section. So yeah, we can make this section a lot easier for ourselves. Looking at this diagram, what we see immediately is that there is this apoptotic stimuli applied to this one cell. It's highlighted there in blue, and that causes this buildup of myosin on this apical basal axis on the neighboring cells. And you can still kind of see the blue cell over there. It's being pushed down towards the basal side, and this is due to apical constriction. As it's pushed down towards the basal side, it undergoes cell extrusion, apoptotic cell extrusion, and that mechanical force, that stress, is applied downwards on it. This eventually leads to folding, as the neighboring cells can then join together. Cool, so what is this happening in? This is in the Drosophila leg joint, so that's covered where? That's covered right here in this paragraph. So more importantly though than the Drosophila leg joint, we understood how exactly it is that apoptotic stimuli can really cause these morphogenetic effects. So let's go to this second paragraph over here. Apoptotic extrusive morphogenetic forces were first reported in vivo for Drosophila dorsal closure. During dorsal closure, the amnioserosa contracts to promote fusion of adjacent lateral epidermal layers. So, okay, what is the amnioserosa there? The amnioserosa is essentially an epithelial band at like the midline of the dorsal side of the Drosophila. So essentially, when the amnioserosa cells undergo apoptosis, as we saw in the picture below, it's a very similar process. So you have the neighboring cells joining together as the apoptotic cells go to the basal side and then, undergo apoptosis. Okay, so let's see what happens after that. They say that, interestingly, genetically inhibiting or enhancing apoptosis delays or accelerates dorsal closure, respectively, with 1/3 of mechanical forces attributed to apoptosis. Okay, that makes sense, nothing too complicated there. In a morphologically similar scenario, mouse neural tube closure requires apoptosis, although causal roles for apoptotic force transmission and cell extrusion remain unproven. So all that's happening here is that in a very similar situation to the Drosophila dorsal closure, you also have a closing of neural tube cells in mice, also due to apoptosis. Um, okay, cool. So we understood that that happens in both Drosophila dorsal closure and mouse neural tube closure. And then in this next paragraph here, again, this goes back to the leg joint development that we saw in the diagram below. But let's again read this paragraph because it does give us some new information that we didn't get from the diagram. They say that extruding apoptotic cells also facilitate epithelial folding in Drosophila leg joint development. Here, sharp discontinuities in DPP activity trigger apoptosis and extrusion, with low DPP cells killed and extruded. So essentially, if a cell does not have that much DPP, then this is a trigger for apoptosis and cell extrusion. And the cell extrusion, they go on to say, happens at the center of the epithelial fold, which causes the folding at the correct leg joint region. They also say that interestingly, sharp boundaries of DPP are also sufficient to trigger live cell extrusion in the Drosophila wing disc. So in the Drosophila fly, if you have a sharp boundary of DPP from one cell to another, then that could actually lead to live cell extrusion instead of apoptotic cell extrusion. So that's an interesting scenario there. Are there any other examples that they give of morphogenetic forces? Okay. They also say that a final example of apoptotic cell extrusion driving morphogenesis occurs during Drosophila metamorphosis, where pupal epidermal cells, termed histoblasts, replace larval epidermal cells. Okay, this is really cool because this is not necessarily a folding scenario or, two neighboring cells connecting and a middle cell undergoing apoptosis, as we saw with like the amniocerosa before. This is the metamorphosis of the Drosophila fly, so you have the LECs completely undergoing apoptosis and being replaced by histoblasts. So again, you have a very similar process happening here though, with the actomyosin rings forming on the apical surface and pushing the apoptotic cells to the basal surface. And these LECs, which are pushed down, then undergo apoptosis and they say are removed by hemocytes, which are Drosophila macrophages. LECs and histoblasts also closely cooperate during tissue remodeling as histoblast proliferation is required for LEC extrusion, and LEC extrusion is necessary for histoblast proliferation. So in other words, histoblast proliferation and LEC extrusion actually go hand in hand. So again, you have this interaction between extrusion, cell extrusion, not just leading to the death of some cells, but also leading to the proliferation and differentiation of other cells. So this really goes to show that the roles of cell extrusion in the body are much more complex than they first seem. Okay, let's head on to the next topic now, which is cell competition and the active extrusion of aberrant cells. Just real quick before we start this, I'll probably break up the analysis of this paper into two videos now because this one's already getting quite long. So in this first we'll just be discussing the positive effects of cell extrusion, and then in the second half we'll address the negative effects. Okay, so another one of the positive effects, as we see here, is that extrusion works hand in hand with cell competition, and this helps to get rid of any aberrant cells, as we see in the title. So let's see how. Actually, take a minute to just read through this yourself. Feel free to pause the video here, and if there are any terms you don't know, go ahead and look it up, and in a couple seconds we'll go through it together. Okay, so what they basically describe here is that neighboring cells have many different interactions, and one such interaction is when you have two cells that compete with each other, and you end up with a winner cell and a losing cell. Unfortunately, losing in rock, paper, scissors is one thing, but losing in a cell competition is something entirely different, because the punishment is cell extrusion, so you die basically. And although this does sound pretty harsh, it does have some really important effects. Getting rid of a loser cell acts as an endogenous tumor suppression mechanism, so it basically gets rid of any emergent cancerous cells that might be acting in a defective or aberrant way. And as shown over here, this happens in a variety of different genetic contexts, and also in a variety of different animals, including flies and vertebrae. This happens also through a variety of different mechanisms, which are detailed in this last line over here. We see that there is a role for junctional remodeling over here. Actomyosin accumulation also plays a role, again. And you also have axon guidance signaling, which also helps to cause upstream cytoskeletal and junctional changes. So in the next couple of sections, I think there's two more. In the two more sections that we have to go, we'll be looking at how cell competition through these pathways and through these different mechanisms helps to eliminate these aberrant cells through cell extrusion. Okay, so now let's move on to the next section here. It's called competition-mediated cell extrusion in Drosophila. So we know from previously in this article that cell competition and cell extrusion go hand in hand. So let's see how that happens in this section. So this does in fact have a diagram, so we can go over that first. And as we see here, when these polarity deficient cells are put in competition with wild type cells, the wild type cells end up being the winners, and the polarity deficient cells end up being the losers. And this caption for part A shows us that these particular polarity deficient cells don't have SCRIB, which is a particular polarity mediating gene, and due to that, it activates the JNK pathway, which then leads to the SLIT-ROBO2 pathway, and this dysregulates E-cadherin. And all of this, simply put, just means that basal extrusion happens, and these polarity deficient cells get taken out. Again, this helps to preserve the wild type cells and make sure that there aren't any aberrant cells in the epithelium, and prevents tumors from forming. As they say in part B here, when the SLIT-ROBO2 pathway is not there, then that means polarity deficient loser cells don't get extruded because E-cadherin is not dysregulated. On the other hand, if there's hyperactivation of SLIT-ROBO2, then you have more extrusion than is needed. And then you get luminal tumors instead of tumors forming in the epithelium itself. Yeah, that's kind of what they show you here. Here you have massive cells in the epithelium, and here you have a massive cells outside the epithelium because they're all being basally extruded. So you don't want either one of those. So because of this, cell competition and cell extrusion have to work together. Now, when we go back to the text, it makes a little more sense. They're basically saying here in the first paragraph, well, actually take a minute to read it, and then we can go over the whole part. Okay, so essentially they're saying in this first section that when a cell does not have the minute protein, or if this is essentially not present in a cell, or has low levels of it, then again it is eliminated based on competition with the wild-type cells in the epithelium. And again, it is then basically extruded because MYC is a polarity protein. It helps to mediate and regulate the polarity. In the second paragraph here, they're telling us something very similar, except this is with the SRC inhibitor, C-terminal SRC kinase, or DCSK. When clones of cells depleted for DCSK are present in the epithelium, then they are exterminated and extruded basally, again because they cannot compete with the wild-type cells, and because you don't really want them to be there, because then you'll have cells that aren't shaped the proper way. Basically, this alters junctional components like E-cadherin, upregulates J and K signaling, and the DCSK mutant cells alter the junctional components like ECAD and upregulate J and K signaling. And if J and K is upregulated, then these are also symptoms that are found in squamous cell carcinoma. So again, you want to have this kind of balance where you don't have too much cell extrusion, but you don't have too little either. That's basically all they're saying in this section. So DCSK mutant cells have to be eliminated. They also add that this is striking, given ECAD's role in transmitting force during apoptotic cell extrusion. Again, this makes sense because if, like in the normal process, as we saw here, E-cadherin is supposed to be dysregulated. But if it's not dysregulated, and it's supposed to transmit force during apoptotic cell extrusion, then obviously this can't really happen if it's not being dysregulated. So then it won't lead to cell extrusion. In this third paragraph, they give us one more example here. They tell us that SCRIB cells, cells mutant for SCRIB, which is a conserved apicobasal polarity gene, they tend to form tumors. And SCRIB proliferation is restricted and eliminated if there are also wild-type cells. Again, the cell competition between neighboring cells and the interactions between them allow this extrusion of the aberrant cells. And yeah, that's basically the point they're making in these 3 first chapters. Sorry, I can't talk today. 3 paragraphs. And it seems like a lot when you first look at it, but if you just kind of break it down, and as I said, if you if you look at the diagrams first, it becomes like a lot easier to understand. That's what I always do. If there's anything I'm stuck on, I just go to the diagrams. So let's keep going then. They have one more paragraph in this section, and they tell us that the slit robo signaling executes squib cell extrusion downstream of J&K signaling. And again, a lot of this can get complicated if you're not really an expert in this particular field. So you want to look for the important points here. So why is it relevant that squib cell extrusion happens downstream of J&K signaling? Well, they actually tell us this in a very straightforward way right here. As J&K signaling is required for diverse phenomena that include cell extrusion, such as cell competition and wound healing, downstream signals like slit robo and subsequent junctional dissolution may more broadly mediate cell extrusion and regulate epithelial homeostasis. Essentially, if we look at this diagram again, just to kind of describe this process, J&K signaling and slit robo 2 are right here. So when they say J&K slit robo signaling is downstream of J&K signaling, it basically just means that it happens after J&K signaling. So here, J&K signaling leads to slit robo 2 activation. And this basically, as they're saying, just adds another layer of regulation to it. So it gives you more control over the regulation of epithelial homeostasis, and helps to more, as they say here, more broadly mediate cell extrusion. So we have one more paragraph or one more section that we're going to go through together in this video. So let's try something a little bit different for this. The title is Competition-Mediated Cell Extrusion in Vertebrates and Cell Cultures. So let's just go through each paragraph and take a minute to read it. And think of one takeaway from that paragraph. And then we'll go through each one together. So let's start with the first one here, and feel free to pause the video. So essentially what they're saying in this paragraph is that competitive cell elimination happens in many different vertebrates as well, and it happens in a similar way to what was seen in the fly findings with the Drosophila. And the majority of these studies have been done with MDCK monolayers and in zebrafish epidermis. So let's take a look at what these studies actually showed in the next paragraph. So take a minute for this one. So the gist of this paragraph is essentially that, like what was seen with the Drosophila fly, when you had squib cell extrusion, it primarily occurred when you had wild-type cells and squib mutant cells that were adjacent to each other, and then the squib cells were extruded apically and underwent apoptosis. And again, this shows the role of squib cell competition in flies and in mammals. So let's go on to the third paragraph now. And the gist of this paragraph is essentially that, again, like what was seen in the Drosophila experiments, squib MDCK cells were also sensitive to mechanical compaction. Essentially, if there was a lot of overcrowding in a specific area, then cell extrusion was able to respond to that and extrude cells that might cause this overcrowding and were, for example, maybe causing a tumor. So this leads to cell competition between a wild-type cell and adjacent aberrant cells as well, and at the same time you have mechanical compaction going on, which cell extrusion would also respond to. And as they say here, moreover, anatom clones expressing oncogenic RAS can compress neighboring wild-type tissue and induce ectopic extrusion up to 3 cell diameters away. So this caused extrusion not just with the immediately adjacent wild-type cell, but even for cells up to 3 cell diameters away. Okay, now let's go on to the next paragraph. So essentially what they're talking about here is the role of oncogenic RAS in cell competition. Here they note that RAS cell competition also involves the S1P and S1P2 extrusion pathway, and S1P2 is required in wild-type cells to extrude RAS cells. Essentially, there's a link here between oncogenic RAS and the pathway that we saw before for cell extrusion, where S1P bound to S1P2. The thing is, the S1P2 receptor is actually on the neighboring cell, not on the aberrant cell itself. So you need the wild-type cell next to the aberrant cell in order to actually cause the cell extrusion. And now let's move on to the last paragraph here, and just do the same thing. Essentially, the point that's being made in this paragraph is that oncogenic SRC cells are very similar to oncogenic RAS cells in how they are apically extruded. And this is independent of cell death in MDCK monolayers. So, yeah, I mean, we just finished going through the gist of this paragraph then, where they basically just outline how competition-mediated cell extrusion in vertebrates is similar to what we saw in the Drosophila fly experiments. And again, it just goes to show that cell extrusion isn't this isolated event in one type of species or in one type of organism, and that it plays important roles in many different in vertebrates, in these Drosophila flies. It plays major roles in all of these types of cells. And yeah, everything we've gone through in this video today shows the positive effects of cell extrusion for epithelial homeostasis. And then in another video, we'll be covering the negative effects, as you can see down here. Let's see, I think I went past it. Yeah, over here, a force for evil, cell extrusion and disease. We'll go over this topic next time. So I hope you'll join me for that video as well. And as I mentioned before, please like this video if it was informational and useful for you, and subscribe to my channel as well. I hope you were able to learn something today, and if you do happen to be an expert in this topic and you just happen to come by this video, then feel free to clarify anything that you feel may need that. Again, I just found this article super interesting, so I decided to go through it today. But yeah, any feedback is appreciated. Thank you, and see you next time.